Okay, welcome back to ImmunoLogic, where immune breakthroughs meet clinical logic, and the no-holds-barred battleground where science takes no prisoners and cutting-edge immunotherapy hits back hard. I'm being very colorful today because we're talking about a rather formidable cancer, brain cancer. So you should know me by now, but just in case you don't, I'm Joe Fraietta, your guide through the wild frontier of innovations in immunotherapy, powered by equal parts of caffeine. Oh, this is water. I thought it was an energy drink. And relentless ambition, at least probably until I hit 60. So today we're diving headfast into the chaos of glioblastoma, and we're really gearing up to beat, as I alluded to, one of cancer's toughest foes. But before we unleash today's deep dive, let me say hello to Janna, my partner in crime, and thanks for leading the episode last time when I was really sleep deprived with Bruce. You did a great job. So are you ready to rock this episode? What do you say?
I'm so excited.
Oh, awesome. Okay, well, today we're really thrilled to welcome a true rock star of cell and gene therapy, and someone, I have to say this, who's not only a pioneer in the field, but also a true mentor, colleague, and I think I can say dear friend, right? And Dr. Marcela Maus. And it's very interesting because when I started at Penn as a postdoc, I was all over the place. I always say, like, I would be nowhere without Marcela, and that is the truth because I had like 8 projects and I don't know what I was doing like day to day. And I really needed mentorship. And she stepped in and just said, you're going to focus and you're going to be laser focused on this one thing and carry it, over the finish line. And she was so right. So thank you for that. And I am eternally grateful. So let me pull up your credentials here. Marcela, if you're in immunotherapy and you don't know who Marcela is, you're like living under a rock. But she is the Paula J. O'Keefe Endowed Chair of the MGH Cancer Center, which I did not know, Marcela. So that must be new. Congratulations. And the director of the Cellular Immunotherapy Program at the Massachusetts General Hospital, and a professor of hematology and oncology at both MGH and Harvard Medical School. And her background is just so extensive. It spans MIT, of course, the University of Pennsylvania. You also did a shorter postdoc at CHOP and clinical training at Memorial Sloan Kettering. And I would say that Marcel's really been rewriting the playbook on cellular immunotherapies, particularly CAR T cell therapy. And her lab is and really has been on the cutting edge of redefining these strategies for cancer, and across many different indications. So from lymphoma to glioblastoma to myeloma, while really mentoring the next crop, the next generation of great scientists. And yes, I have to say, balancing life as a mother to 3, along with a large dog and even a gecko, right? So I picked that up last night. So, Marcela, it's great to see you. Welcome to ImmunoLogic, and let's get into the thick of it. And we're excited to pick your brain, pun intended.
Thank you so much, Joe and Janna. It's such a pleasure to be here. And all of your kind words, Joe, just really warm my heart. It's been great to see you grow as well.
Oh, thank you. Thank you. Well, if I may, let's jump right in. Jana, is it okay if I start? I was thinking about how to frame this episode, and like I said, there are so many things that we could talk about. You've done so many wonderful things in the field, but this week I had a distant cousin, and she called me and she said, my father-in-law unfortunately was diagnosed with glioblastoma, and I've been reading all about it. And what can you tell me about cell therapy? And this paper came up in the New England Journal of Medicine by Marcela Maus, and it looks really, really innovative. And so that was, really the inspiration for this episode. And so I think it was last April, you and your team reported striking results using intraventricular, what you call CAR-V3 T cells in patients with recurrent glioblastoma. So, Marcela, would you mind just breaking down the study a little bit for us? Like, what are the key takeaways and what makes these CAR T cells uniquely suited for doing battle with glioblastoma, if you will.
Sure. Thank you so much, Joe. Well, I'd like to, if you'll grant me a few minutes to talk a little bit about the history of this, about how we got to this particular study, and then talk about the study itself. So, the story goes back actually to when the first CAR T cells were really starting to work in leukemia and lymphoma, which was work being done at Penn, circa 2011, 2012. And at the time, we thought that if we could just change the target antigen of a chimeric antigen receptor, that we would be able to have these dramatic effects in solid tumors. And so we embarked on a collaborative study with Penn and with Pittsburgh at the time. Hideo Okada was there. And we developed this CAR targeting this mutated antigen that occurs in about 20% of patients with glioblastoma. It's called EGFR variant 3. And we thought that we would just be able to give a single dose of these CAR T cells targeting EGFR V3 into the arm and get anti-tumor effects. And we sort of rapidly did the preclinical work and took it to a clinical trial. We published the preclinical paper showing that that CAR had really strong effects in small animal models bearing human glioma, and it was looking fantastic. We were really focused on targeting just that mutated antigen. We took that to clinical trial. We treated 10 patients, and really there wasn't a lot that happened with anti-tumor response or with toxicity, but we were really privileged to work with a neurosurgeon, Don O'Rourke at the time. He did biopsies and resections of those tumors that were remaining, and we learned that the CAR Ts could actually get into brain tumor, which is a really big issue in brain tumor because it sits behind the blood-brain barrier, and there's not a lot of drugs that get into the tissue. So even if you give an antibody or even a chemotherapy IV, it just doesn't get into the brain tumor. That's been this huge issue. And so T cells, even though we give them into the vein, they got into the brain tumor. But we learned a lot from that trial in that, there was this adaptive immunosuppressive response, and also that V3 was just not a high enough antigen expression. There was a lot of EGFR there, but not a lot of V3. And so now we're back to 10 years ago or so. So I really wanted to be able to target all that wild type EGFR, which is not on normal brain tissue, but it is in almost all brain tumors. And so we built on that CAR, then having it secrete a T cell engaging antibody molecule, which we call the CAR team. And the idea is that now we have a CAR working as a team with a T cell engaging antibody. And so that's why it's CAR V3 Team E. One of the other things we learned is that giving CAR T cells into the vein, we could get some into the tissue, but we really didn't know if that was enough, if there were enough of them in the tissue. And so we decided, again, building on work that had been going on in the field, particularly at City of Hope with Christine Brown and Behnam Beatty, but it was possible to give CAR T cells into the ventricle, which is this reservoir of fluid that bathes the brain. That's where the fluid is made and it sort of circulates around the brain. So if we could get the T cells there, we might be able to get more of them into the tissue. And so when in my lab at MGH, when Brian Choi was a fellow in my lab doing his research years during his neurosurgery training, we did those experiments in small animal models. And then eventually, as time went on, Dr. Choi now became a neurosurgery assistant professor, which he's at Mass General now. And in the meantime, we opened a study. And there was so much innovation here that it took us a little bit longer to get into our first in human because we were giving them through a device into the ventricle. We were having the CAR make a second drug, and so that required sort of a specialized set of tests. So when we treated our first patient, first visit, we were all really unsure of what would happen, and it was dramatic. We didn't, we were nervous, but it was dramatic in the sense that 24 hours after we gave the infusion, the patients had, fever and altered mental status. They were sleepy. And so that's why we ended up getting scans because we hadn't really planned to get scanned so early on. And so we got these scans, and really to our great surprise, it looked like the tumor was getting smaller. And that's not something that we see in glioblastoma. So,
Right.
I'm not a neuroscientist. I don't read MRI of the brain as my clinical specialty, but everyone was just super excited. In fact, excited and skeptical. And so we redid the scans like every other day for the first week just to really confirm that there wasn't some artifact or something, and it wasn't. And this was something that was consistent. But what we really want, of course, is for those responses to last a longer time.
Wow. So that must have been such an aha moment. And it reminds me of the days of, CD19 CAR T cells, right? When they saw like clear bone marrow. uh,
Yeah. And it was
and they were like,
repeated.
and yeah, and it was like repeated. They didn't believe it. So that must have been something. So you scanned just based on toxicity and you
Mhm.
saw, wow. So
I mean,
Yeah, but stand
I'm
really
sure
based
you might be
on.
the same way, Marcella, but if I can see a big change on MRI, then you know it must be a really big one because my MRI reading skills are not the same as other imaging modalities, that's for sure.
Exactly, exactly. So that's just been it was unexpected. And obviously the first time we saw it, we were scanning because we were worried that we had done something that could be toxic. And but then it looked like the patient was started to get better quite soon after that, and the imaging stayed. Um, there was like confirmation that the scan was really changing based on the therapy that we had administered. And um, I would say we're optimistic, but we also are a little bit cautious. Like, glioblastoma has been generally a very difficult disease. Prognosis can be relatively poor, and so it remains to be seen whether this will like really, change the clinical outcome for a lot of people.
So what about readministration of the product? Is that possible? And then my other question is, and it's been a year since I've read this. I was like looking through the figures again last night so that I would be prepared for this. But um, I guess when there was disease progression, did you have an opportunity to look at the tumor that was sort of resected? And in terms of, I guess, just cells and antigen expression, did you see antigen? Because it leads into, is targeting two antigens enough? But with this approach, really, you're just pulling in, T cells. You could just, right,
Yeah.
by virtue of the engager. So what do you think? Is two enough, or do we still not know? Can we read a
Yeah.
lot of questions
I
there?
mean, there's so many questions, Joe. So we're targeting like two antigens in the same family. So I
Right.
would say we're targeting two antigens, but they're kind of part of a family. They're not like two completely unrelated separate antigens. Um, and we don't have, at this point, we don't have a lot of data on, what kind of tumor is recurring and whether it's antigen loss or not. Um, obviously getting a brain surgery is kind of a big deal. And so we
Yeah.
don't routinely get that. And so it has to be sort of the right thing for the patient at the time. We don't do brain surgery as like as a research activity just because it's too
Right.
high risk, right? So it has to be what the patient needs. And then if there's tissue that we can look at, then we do that. And so that opportunity may present itself in the future, but in that first paper, that hadn't happened yet. Um, the question of whether targeting one agent is enough or not, I think is still to be determined. I think what I'm really excited about is that we seem to be changing the biology of the brain tumor, and that T cells are getting in there, and we are seeing effects that are strong enough to be visible on imaging. Whereas in our previous experience, for example, when it was given IV, we saw some effects of the CAR T cells, but only in tissue that was resected. We didn't really see any significant changes in imaging. And so
Mhm.
that's just telling us that we're getting like a pretty big signal of stuff that is happening in the tumor. And so I think that gives us something to build on.
Absolutely.
It's really interesting to me too that and exciting that the EGFR targeted team secretion doesn't seem to have any systemic toxicity. Um, has that emboldened you to target alternative antigens in the CNS that would otherwise be uh, be deemed just too risky?
Yeah, that's a great question. So, uh, I mean, I'm still sometimes surprised at uh, the risks that we take. I feel like it was really quite nerve-wracking just to do this one. I'm not necessarily the one who's going to be quite ready to do that, many other high-risk antigens, but one step at a time. And when we get a really positive signal like that, then it sort of does embolden you. We're trying to carry on this study in larger groups of patients, and we've added some interventions to try to enhance the persistence of the CAR Ts, because what we saw in these first patients where because we were giving the cells into the CSF, we initially did not plan to do any lymphodepleting chemotherapy or preparative regimens. And, that has been necessary for every other CAR T to work in heme malignancy and other solid tumors. And so, but because we were giving it directly into the CSF, we thought we wouldn't need it initially. But based on what we're seeing in terms of the expansion and the persistence of the cells in both the brain and in peripheral blood, we realized that we would actually may want to have some chemotherapy to try to enhance that persistence. We do see the cells initially, they're just in the CSF, but they do actually like poke out into peripheral blood where they're detectable about 2 weeks later. And then they went away. So it was very transient. And so the next question really is going to be, as we're adding lymphodepleting chemotherapy, do we still maintain that sort of that therapeutic window and that safety profile where we're getting more expansion in CSF, which is what we want, and yet not having it have any on-target off-tumor toxicity in the systemic circulation?
Are you
So were
familiar?
you at the, oh, go ahead. Sorry.
No, you go, Joe.
No, are you at the optimal dose, or is there any room for further dose escalation? Probably not, right? Given, I guess, the potency.
Yeah, I think, yeah, that's a great question. I think, from a relative to what we dose in heme malignancies or when we've given cells into the vein, we're so much lower, right? We do one manufacturing run and we can get 6 doses out of that, like 5 or 6 doses, because we're really only giving, 10 million viable CAR T cells into the CSF. And, for context, as you know, like most of the others are more in the order of 100 to 300, even 500 million cells.
Yeah.
So we're pretty, like our dose is pretty low. So the question is, can we dose escalate? And I think, you know, when that first patient was pretty sick, and what we saw in the next two patients also who initially had these high fevers and sleepiness, I would be a little bit nervous about increasing that dose. I think really what I'd like to increase is the persistence and like sort of the area under the curve, but not necessarily all at once in the first hour. So or the first 8 hours. So what I would love to be able to do is like redose or give lymphodepleting chemotherapy so that we get to like a more persistent state. And then, if we needed to redose later, we could do that. When we initially wrote the study, it was meant to be like weekly dosing, because that's what we thought we'd need. But then when we saw the data coming back on PK, we realized like we don't need weekly dosing because there's still tons of cells there.
Right.
What we
Was that
really
the amended workflow, if I remember?
Yeah, so that was a little bit different, because this was a safety run-in arm where we were just going to give one dose and follow. And I'm so glad that we did that rather than like immediately going and I credit FDA for suggesting strongly that we do that. I would say, so the amended workflow, initially we wanted the recurrence. So this trial is open for patients with first recurrence. So they've had their initial presentation, their diagnosis, their resection of the tumor followed by radiation, and then it has to look like the tumor is coming back. And so we initially thought that we would need the detection of the antigen, EGFR variant 3, as well as EGFR on that recurrent tumor. But that introduced a logistical problem, which is that they would need surgery to obtain the recurrent tumor and then sending it for pathology, getting the testing, and then we would initiate sort of manufacturing and prepare the product. And the patients just don't have that much time. And it would require another surgical procedure then to place the device to deliver the cells. And so it was 2 surgeries and a longer wait time in between. And given the pace of this disease and the lack of alternative efficacious therapies, we consolidated it and changed the workflow so that we were using the the tissue from the initial resection from first presentation to determine eligibility in terms of EGFR-V3 and EGFR expression. And then when the patients have their first recurrence, we could do one surgery where I say we as if I'm doing it. It's not. This is Dr. Choi and all of his neurosurgical colleagues, where they resect some of the tumor. They sample the tumor so that we get the data about the antigen levels, but we get that later. And they also place the device at the time so that they have this little catheter that goes into the ventricle so that we can just infuse the cells at the bedside by putting just a small needle into the scalp. Patients like awake and talking and sitting up in bed when we do this.
Got it.
So the workflow was really to try to overcome those timelines and logistics about how many surgeries the patients get and the time from when they become eligible to when they can actually get the product.
But so much of that is just great intuition. So I love that about this study that, you had these really striking results and you just have to sort of evolve almost in real time, right? And I'm not sure many centers are great at doing that. So it really speaks to your expertise and yeah.
Well, it definitely
practical and patient-centered study.
Yeah.
Yeah, I mean, it really takes a team of people, right? So, and listening to each other. So when we hear this about from pathology and we hear this from neuro-oncology and we hear this, from the patients, like and we're counting the days and they're counting the days and it's like, okay, we gotta, after our first initial thoughts, we're like, okay, we gotta make this realistic. And I will say it's also sort of a plug for doing this, as investigator initiated like academic studies, where we do have some flexibility. We do this within the regulatory framework, of course. We have, protocols and minutes and, discussions with regulatory authorities, but we don't open 5 sites at once and we don't, we're not treating, we have a long stagger in between patients, which is both good and bad. The good is that we can maintain some of that flexibility and we learn from each patient and to try to improve our process. The bad is that we have so many, there's two really. One is that we have so many patients who want to participate and such limited numbers of slots, right? So that's really difficult. And with a disease like glioblastoma, it's been really difficult to even have like a long wait list because you can't have someone say like, okay, we can get this therapy in 6 months because things may really change in that amount of time. So that's a challenge. And of course, it will just take us longer to get to like a statistically meaningful number of patients and to get to the kinds of endpoints that one would need to really know whether to go onto phase 2 and so on. So there are definitely limitations to what we can do in academia, but I've really enjoyed and think that it's a very viable path to do some of that initial sort of I don't even want to call it exactly dose finding, but just to have that first in human experience with an investigator initiated studies, I think is really useful.
Yeah, tremendously valuable.
Mm-hmm.
So Janna, how about your dream big question? This is, we ask.
Okay, if I have another couple small ones.
Oh, go ahead. Yeah.
So one was, I think that the third patient that was reported in that paper, in retrospect, had lost EGFR V3. Do I remember that correctly?
Yeah.
And yet still appeared to have a response. Do you think that there's any role for an edited T cell that is just edited to express this team? Like, do you think that patient, it was kind of sampling and there's heterogeneity in GBM and she actually had EGFR V3 expression? Or do you think that all you needed was some T cells, and this team molecule?
Yeah. So probably some element of both. So it's really hard to know what happens with such small numbers of patients. But EGFR V3 can be a splice variant. So when there's amplification of EGFR, it's likely that there is some V3, and it's hard to measure at any snapshot in time to know exactly how much there is and on a per cell basis. But I think it's also probably true that just targeting EGFR will have some effects. And I say that based on our preclinical data where we had some tumors that were engineered to express EGFR. And just by having the CAR team injected into those small animals, we were able to get anti-tumor responses in 80% of the time. So I do think that there is some effect there. I think that having the CAR there is still actually essential because of the co-stimulatory molecules embedded in there. If you just have a T cell that secretes the T cell engager, you're not getting the benefits of the co-stimulation. And so, especially for the persistence that we're trying to engineer and that sort of deeper expansion, I think that the CAR part is actually essential. It doesn't necessarily have to be that target antigen. That's what we have, but it could be something else. But I do think that the combination is a really useful design.
Well, we only have a minute or so left to pick your brain. Would you perhaps tell us what your most controversial opinion in cell and gene therapy is?
Oh gosh, my most controversial opinion is probably like autologous versus allo or in vivo or something like that. Um, I'm a strong believer in autologous. I feel like it's already really hard to engineer the body and engineer biologic agents without running into issues. And I think autologous is the way to go. I think, with allogeneic, you're just swimming upstream in terms of like organ transplants really hard, bone marrow transplants really hard. Like I think it's a fallacy to think that we're just going to overcome it, with the snap of the fingers and make allo cells. And then with in vivo, I think it's got some possibilities, but I think it's going to be hard. Like systemic gene therapy has also been really hard for, decades. And so I think the idea that you could just like snap your fingers and then it will become easy and will supplant autologous therapies. I'm not. It's not that I don't think it will ever work. I just don't think it's going to be easy.
You can tell that Marcella trained me. I'm of the exact same mindset. But it often gets me into trouble when I say like autologous.
Well, I think, honestly, people who do a lot of human immunology can probably wrap, like, that's the perspective, right?
Right.
Yeah.
It's true. So thank you, Marcella, for sharing really these deep insights. And this was a great episode. It's really encouraging and also sobering to see how much progress has been made. Also, how much work remains to really refine these therapies for clinical use, especially in this disease. So, with that, that wraps up today's deep dive on ImmunoLogic. To our clinical colleagues and researchers, thank you for joining us as we navigate the frontier of cancer immunotherapy. And as breakthroughs continue to evolve from the bench to the bedside, remember that every new insight brings us one step closer to redefining treatment paradigms, hopefully. That's why we're, in this and we're all in it together. So stay tuned for more groundbreaking discussions. And until next time, keep challenging the status quo and pushing the boundaries of what's possible in cancer immunotherapy.